this series of videos on immune responses, we've been focusing on the different cells and proteins that we have in our immune system to fight our pathogens. So what we're looking at today is the concept of immunity, which is basically preparing your body enough so that it has adequate defenses against pathogens. Immunity is all about antibodies. It's either the development of antibodies upon exposure to an antigen, or we're looking at short-term transmission of an antibody to help us deal with the pathogen really quickly, but not actually gain long-term memory. When we look at immunity, we've got a few different things we can have. We can have either artificial versus natural immunity, or we could have passive versus active immunity. We're going to be looking at the combination of these things to give us the 4 different types of immunity. Let's first look at the distinction between passive and active immunity. Active immunity is all about producing memory cells. We're making immunological memory by producing memory T and memory B cells. So we can get this either naturally from normal exposure to a pathogen, or we can get this artificially through injections or inoculation with a part of the pathogen or the whole pathogen itself. Passive immunity, on the other hand, is a short-term measure of just antibodies, and we do not form memory cells. In our previous videos, we've essentially looked at all the different steps involved in developing natural active immunity. So this is upon normal exposure to a pathogen. So someone got sick and they coughed in your face, which gross, disgusting, don't do that. But that transmitted the pathogen to you. You developed a long-term memory from your immune system working and doing its thing, and we have memory T cells and B cells in reserve for the future. Because of this, we have natural transmission of the antigen, and we have active immunity because we do have memory cells. Artificial active immunity, however, is all about vaccines, which is taking a bit of the pathogen or the pathogen itself, introducing it into your body in the hopes of triggering an immune response where we produce those memory B and T cells. The very first vaccine was created in 1796 in response to this disease called smallpox. Smallpox is a viral infection. It causes really extensive rashes and lesions, and eventually the immune system collapse. It was basically ravaging Europe for quite a while. So it was a very, very serious disease. Even royalty got it. King Louis XV of France died from smallpox. So everyone was affected by it, and no one was immune because of class or wealth or anything like that. There were some treatments for smallpox, but they weren't very effective, and some of them were quite ill-founded. So there's evidence from the 15th century Chinese medicine existing where someone took a dried scab of someone with smallpox, dried it up, ground it up, and then basically told people to snort it. So, people tried. But this disease was way more serious than they had defenses against. Now, Edward Jenner, who was an English doctor in the late 1700s, he made an observation. He noticed that local milkmaids were milking cows and they were getting this disease called cowpox in their hands. So that was a much less serious disease than mild rashes, but that's basically it. They got cowpox, and he noticed that none of the milkmaids ever developed smallpox. And he hypothesized that there was a connection between these two viruses, that something in cowpox can confer some form of protection against smallpox. His hypothesis led him to do something that today would be super unethical and not okay at all. But back in the day, people just didn't seem to care. So he went straight to human testing. Jenner took the pustules of a cow that was infected by cowpox. He took a scraping from that pus, and he infected a small 8-year-old boy named James Phipps. He cut him and he put a bit of the pustule into the cut, and he just waited. Phipps did develop cowpox, and this is the bit where it's just don't do this, please. He took James Phipps and then he infected him with smallpox, and he was very lucky that James Phipps did not develop smallpox. Super, super, super lucky. But he did work out that, there is a link between these two viruses, and he gave our first ever vaccination. Vaccinations since then have obviously become much, much safer. Don't do this at all. Whilst Jenner was extremely lucky to have actually created the world's first vaccine and not get sued by the Fitch family, people were obviously a bit hesitant about this as a treatment. They said that if you got the cowpox into your system, you'd turn into a cow, and there were like lots of political cartoons about this. Jenner was ridiculed, but he went on with his work anyway. In fact, the word vaccine comes from the word vacca, which means cow, so it's a little bit of like a nod to the original vaccine. The vaccine against smallpox was more widely accepted over time, and since then we've obviously developed way safer vaccines for smallpox. In fact, we developed such an effective vaccine against smallpox that we consider the disease eradicated in the 1980s because of such widespread vaccinations around the world that the virus didn't really have natural hosts to live in anymore. And so the only sort of copies of the smallpox virus that we have now live in very secure labs. Vaccines are all about artificially introducing a bit of the pathogen into your system so that you do produce an active response and form long-term memory. We can induce artificial active immunity by a few different ways. We can either use a weakened version of the pathogen, which is known as an attenuated vaccine, or we can use a version of the pathogen that cannot replicate anymore. So this is called an inactive vaccine. Finally, we can also just introduce an antigen from the pathogen. This is known as a subunit vaccine. Introducing a vaccine into your system is usually enough to trigger an immune response against the antigens from our pathogen. So we can look at like a virus or bacterial cell. When we have those memory cells in reserve, the next time you encounter the pathogen in your day-to-day life, we can launch a really big attack of antibodies that will take care of the pathogen before you see symptoms. So if you look at this graph of antibody levels in response to primary and secondary exposure to an antigen, we can see that the first time you encounter the antigen, it takes a little while to produce antibodies, and eventually that antibody level will drop. The initial exposure is the equivalent of you introducing the pathogen through a vaccine and you launching an immune response to create those memory cells. Upon second exposure to the pathogen, so this is like you in the day-to-day life come in contact with the pathogen, we have a very rapid response producing a large number of antibodies, and this will deal with the pathogen super quickly that you might not see symptoms. Both natural and artificial active immunity will get you to the same point. So we are having a protection in our body that's going to lay in wait for the long term so that you have something to help you defend yourself the next time you encounter a pathogen. Vaccines are safe for use in the general population. So if you've got a normal immune system, you should be able to deal with a weakened version of the pathogen and not have any problems. There are some people who cannot have vaccinations, so they might be allergic to the ingredients in the vaccine. They might be too young, too old, or they might be immunocompromised, so they might have cancer, HIV, or they could have some sort of disease that's stopping their immune system from working as it should. In that case, what we rely on to keep those immunocompromised people safe is herd immunity. Herd immunity relies on a particular percentage of the population being vaccinated so that we can protect those who cannot get vaccines. So we're looking at making sure that if a virus, let's say, enters my body, I'm vaccinated so that I cannot pass this virus onto another person who is immunocompromised. The disease stops at me. For herd immunity to be effective, we need high percentages of people being vaccinated. So we're looking at percentages in the high 90s. In that case, a disease will not outbreak. It will not spread from person to person very effectively, and we can contain the disease and not see those who are very vulnerable getting sick. As the percentage of people in the population without vaccinations increases, however, we actually have a large number of people where the disease can be transmitted between, and we have an increase of the disease in the population. We're leaving those more vulnerable, basically really on their own. We don't have the rest of the community working together to keep them safe. At the heart of it, herd immunity is an altruistic concept. It is about protecting those who cannot get a vaccination, those who are much more vulnerable in the population. So it is not just about your own protection, it's about the protection of others as well. Obviously, we can't talk about vaccines without talking about the anti-vaccination debate, which I use that term very, very loosely. The argument against vaccines, because of its supposed link between autism and getting a vaccination, all stems from one paper published in 1998 by a former gastroenterologist named Dr. Andrew Wakefield. He said that there was some link between the mumps, measles, and rubella vaccine and autism. Now, his work was widely discredited. We found out that he falsified all of his data, he made it all up, and everything was retracted. However, because that paper was published back in the day, people latch onto it and use it as an argument, and they don't pay attention to so much research that's been done since then that says, no, he made it up. Vaccines over time will give you long-term immunity, but for some vaccines, this effect does wear off over time, and we're kind of not really sure why that happens. In the case of a vaccine wearing off in effectiveness over time, what you might get are booster shots, so you might have another dose of the vaccine over a few years, and that will increase the numbers of memory B cells in reserve. And from that you get long-term immunity for the rest of your life. You get protection. That's a good thing. Unlike active immunity, passive immunity is all about short-term gains without long-term immunological memory. So we're looking at taking antibodies from someone who has actually produced antibodies against a pathogen and taking those antibodies, introducing it into someone's system so that they have a short-term treatment but they don't produce those memory B cells. In passive immunity, we also have artificial or natural immunity, and this just all comes down to where we source the antibodies that we are going to administer to a person. If we look at natural passive immunity, we're looking at antibodies made from a person and passed to another person through processes like breastfeeding. A mother will have her own antibodies against whatever pathogens she's encountered in her lifetime, and she can transfer this to her child through pregnancy. So during pregnancy, whilst the baby is inside of her body, she produces some antibodies that will cross the placenta into the baby and produce a nice protective sort of barrier. Natural passive immunity is also found in breast milk secretions. So as a mother produces breast milk, her own antibodies are produced and go into the breast milk, pass onto her child, and protects the child for as long as they're feeding. The antibodies we tend to see in breast milk tend to be like IgG or IgA, and they'll give a nice short-term protection but no memory cells. So when the baby stops feeding, the protection stops and the baby does not form any long-term memory against those pathogens. Artificial passive immunity is all about antibody serums. So to make an antibody serum, what we need to do is we need to infect someone or an animal with the pathogen. They produce antibodies because their immune system works against it, and we are going to purify these antibodies from their blood plasma. When we have this, we essentially have an injection of antibodies that we can give someone as a short-term defense against the pathogen. And so we're basically making it so that their immune system has time to catch up and treat the pathogen. Their immune system's not producing those memory cells or anything like that in the long run, but we have defenses to deal with the pathogen really, really quickly as needed. We can make antibody serums against things like toxins, for example. So if you get a snake bite, we have a reserve of antibody serums that are specific to the antigens that we have in the venom from a snake bite. So if you get a snake bite, we take what we call an antitoxin, which is an injection of those antibodies into your system. They will bind to the toxin from the venom and it no longer affects your system. Another way of conferring artificial passive immunity is through a plasma transfer. So right now with COVID-19, what we're looking at as a potential treatment is taking the plasma of those who have previously been infected with COVID-19 and transferring it to people who are extremely sick with an infection and it doesn't look like their immune system's catching up very well. So we take this plasma which contains the antibodies, we give it to the person who is sick, and we hope that's enough of a treatment so that their immune system can then go on and fight the pathogen. Another way to produce antibodies for artificial passive immunity is through making monoclonal antibodies. Monoclonal antibodies are lab-derived antibodies. To make a monoclonal antibody, it is a bit of a fiddly process. It does require quite a few steps, and it requires an animal host as well as a human cell. So to do this, what we need to do is we need to identify what antigen we want to target. Once we have that antigen, we can then introduce it to the system of an animal. So let's say we're introducing it to a mouse. The mouse is going to launch an immune attack against that antigen, and we're going to harvest their spleen cells. The spleen is one of the organs of your lymphatic system, so we've got cells that are capable of making antibodies in the spleen. What we're going to do is we're going to take those cells and we're going to fuse those cells with a human cancer cell. We call this a myeloma cell. Now the benefits of this is that a cancer cell is able to replicate again and again and again without dying. And because it's a human cell, we're not going to reject anything that's being made from this human cell. So we're going to fuse this human myeloma with the spleen cells, and we create this cell called a hybridoma. It's a combination of the animal as well as a human cell. The hybridoma cell is going to replicate again and again and again, and as it replicates, it's producing a bunch of antibodies. The antibodies we now produce are going to be a combination of human genes that produces protein, so the majority of it is human, and at the very end, the variable region will come from the mouse that was infected by the antigen. So the variable region is going to be specific against the antigen we're targeting. These are a monoclonal antibody. They come from one source and they're all identical. We can harvest these and purify them, and now we can introduce them into our system as a treatment. Monoclonal antibodies made against cancer cells will have a variable region that is complementary to our cancer antigen. When their monoclonal antibody sticks onto their cancer cell, we can also have it deliver some drugs against the cancer. So we can have drugs at the end of the monoclonal antibody, generally in the constant region, and when it binds onto the cancer cell, it is going to deliver this dose of this drug and it will kill that specific cell. We can also have it hold something that has essentially radiation and deliver some radiotherapy that way as well. As a treatment, we can use our monoclonal antibodies to target really specific cancer cell in a patient's body, which is a downside of like normal chemotherapy where we might attack healthy cells as well. But in this process, it is very targeted to the cancer cell, and we tailor make this for each person's body. Obviously, as a treatment, it is a passive process. We aren't immune to cancer. We are not going to be immune to every single cancer that comes out in the future because they might have different antigens. But as a short-term treatment, it is very, very effective. Overall, I hope you've learnt the difference between passive and active immunity and how it can be naturally or artificially acquired. These are all things that we have to boost our own immune responses, and hopefully we have some protection against pathogens in the future. If you don't have protection against pathogens from natural exposure to the pathogen, we are going to rely on vaccines. So please get vaccines, vaccinate your children, because without it, people are going to die. You know, the world right now in lockdown because of COVID-19, this is a world without one vaccine. So imagine what the world will look like if we didn't have all the other vaccinations available to us. Yeah, we're going to die even faster. So please, vaccinate yourselves, vaccinate your children. Seriously, do it.